Okay, students, it's time to talk about experimental designs some more, particularly single factor designs. So let me just ask you, what is a factor? You can pause and try to answer that question. What is a factor? So the single factor design is the single factor. It refers to a single independent variable. If you pop into the next chapter after this, we are going to look at factorial models and factorial. What is this chapter? Factorial designs. Okay, so this is really building knowledge that you are going to need for next chapter. And so it's good to make sure that you master all of these things before we move on. First of all, with a single factor model or design, you can have two or more levels, right? The minimum would be two levels because you need an experimental and a control group or something like that. So you need two levels to compare, but you can also have multiple levels. Now, I don't always like these little um, figures that they put into the book, but on page 217, figure 7.1, I think that's a pretty nice summarizing figure as to where we are at so far. Now, when you look at that and when you get into this reading, you realize that there are a few new terms that the authors are introducing. And I would like to tell you that there is sensibility behind this. But I think what they're just bringing up is that some of these particular methodologies um, have multiple names. So sometimes within groups design is also called a repeated measures design. So you just need to know that. A subject variable design is also called an ex post facto, which means after the fact. So we don't divide these groups. We look at these groups after the fact, which is after the fact that they're already divided. They're subject variables. Okay. So, and then just with a normal between groups design, we're going to call that an independent groups design. It can also be called an independent groups design, which actually makes some sense that you are randomly assigning these groups into independent groups. Okay, so that's one way to think about it. Now, beside it, when I have this figure out and I'm looking at this kind of hierarchy of are all the kind of single factor design options. It comes down to four of them, but I also want to draw your attention to the inferential statistic that is used for each of them. Okay. So um, the inferential statistic for any kind of single factor design is a t-test. And a t-test just tells us simply, is there a statistically significant difference between our two samples? So if we were just looking at our two samples, right, descriptive statistics, we could tell if there's a difference in the dependent variable for with both of them. But then we use the t-test, which is the specific inferential statistic for single factor designs where there's just two levels. Okay. We're going to use that t-test, and that's going to tell us whether we can generalize our findings to the population. Obviously, this would only be generalizable in within group design, where you're comparing a group against themselves, counterbalanced as a control, or an independent groups where you are randomly assigning groups, or a matched sample. Okay, a matched group. What it won't work for, as you know, is an ex post facto or subject variable single factor design. Okay, how much of that did you catch? Hopefully that makes sense. Okay, so t-tests are used to tell us, is there a significant enough difference I should say that again. Is there a statistically significant difference that we're pretty sure that the effect that we're seeing here is generalizable? And one other thing I just want to draw your attention to is that we have independent t-tests and dependent t-tests, and they're just you just have to learn them. You don't have to understand them at this point. You just have to know that there is that an independent t-test is appropriate to use sometimes, and a dependent t-test is appropriate to use sometimes. The good news is that they just that never changes. So you should always use an independent t-test for a between groups design for independent groups. Okay, so independent groups, where you're using random assignment, you're going to want to use an independent t-test. Also, you use an independent t-test with ex post facto or subject variables. So in those cases, use an independent t-test. The dependent t-test is used for matched or repeated measures designs, and then also, I'm sorry, a matched group design or repeated measures design. And however you need to learn it, I always tend to think that, you know, if it's in a matched group, my score is really dependent and being compared to somebody else's score who I'm matched with. And so that's how I think about dependent. And I also think about repeated measures. I think about how I am dependent on my own, my score previously. And so there's a dependency there. That's how I remember it. It's probably not even statistically correct what I'm saying, but that's how I remember it, is that those two scenarios have more dependency, and so that's the dependent t-test. Okay, so that's important to make a note to. All that to say, on that figure on page 217, I definitely have, beside independent groups, one factor design, I put independent t-test, and then dependent t-test, independent t-test, and dependent t-test across the page. Okay, all right. Wow, I don't know how much I'm going to be able to help with particular things until we get into the questions themselves. And so, really important that we are going to be meeting this week, and that you use Ask Questions here. The truth is that if you have been hanging in there so far and are really doing the work and understanding things to this point, this chapter is not going to be too difficult for you. Now, if there is more than than two levels of the single factor or the independent variable, we don't use a t-test now. We use an ANOVA test, okay? And an ANOVA test is going to let us know whether one of the conditions, so in this case, we'd have three conditions if we are measuring caffeine consumption and its effect on rat performance on a maze or something like that. It's such a boring example, I know, but so if we give them like 0, 4, and 8 milligrams of caffeine, what an ANOVA will do for us is let us know that one of those three conditions is or is not statistically different from the others. And then ANOVA kind of flags it and says, you've got something here, but it doesn't tell you exactly where. And so after that, once you run an ANOVA, then you can do additional statistical testing to find out exactly which level of your independent variable was different, okay? And just like there were independent and dependent t-tests, ANOVAs as an inferential statistic also are independent and dependent according to the same pattern, okay? So for example, if you are doing a between groups design, then you are going to do an independent ANOVA if you have 3 or more levels. Think about this though, like even if you had 6 levels of your independent variable, so let's say that we were going to do 0 milligrams of caffeine and then 1, 2, 3, 4, 5 milligrams of caffeine. If you did something like that, an ANOVA would tell you that at least one of your conditions is statistically significant. It can be generalized. It's a great thing. And then you would do more post hoc testing afterwards, statistical testing, to find out exactly what happened. But it flags it. it's very helpful in this way. Um, in well, with your null hypothesis being that there is going to be no effect, the ANOVA will tell you very quickly whether you can accept the null hypothesis. Because if there is no statistically significant ANOVA, then you must accept the null. Okay. Anyway, so hopefully that's helpful. This chapter gets into a little bit of graphing. It's really important that we can take statistics from research and that we can put them into figures that convey a lot of the meaning in graphic form rather than written form. Think about it. If you've ever seen a graph that you understand, it's a much quicker depiction of what's going on than reading a paragraph with a bunch of numbers in it, right? So you can see immediately. Now, one of the things that we have to be careful of is how we represent these, that we represent them appropriately. This is the validity of using proper techniques when we are representing data, but also the types of variables that we are using are going to indicate whether we should do line graphs or whether we should do bar graphs. So for continuous variables, we want to do line graphs. Okay, so think about think about giving that 0, 1, 2, 3, 4, 5 milligrams. This would be a good line graph because we have these intermittent this consistent numbering, right, where the number between 1 and 3 on this graph is 2. It's like a ratio, right? So don't get that confused with ratio scales for dependent variables, but the independent variable can be a ratio or a continuous variable as well. Okay, so yeah, there's some really interesting things in there, but I think they're pretty straightforward about when you should be using what kind of graph. Let me think about this for a moment, because somebody asked me a question about interpolating or interpoles, which is really just this idea that when you have a line graph and it is saying on a continuous variable, from 1 to 10, if it goes from 1 here to 10 up here, if you draw that line straight across, you are assuming that 5, that interpole of 5, which doesn't exist on that line, but that interpole of 5 is actually on the graph where you are saying it is. And this is why it is good to have more data points on a graph. Now, interpolation is really great when you have this happens with correlation a lot, where you can predict with one variable, you can predict where the other one will be. Now that's you use interpolation in that situation because the regression line, and we'll get into this in a couple weeks. That, for example, this score on this variable generally tends to be correlated with this score on this variable. Okay, you've got that point on the graph. Okay, what we're showing here is that you can't always be sure. It's always better to have more data points to see what the trend is. Okay, so yeah, I think there was an example about caffeine. Was it caffeine? Oh, I can't remember. Yes, I am on. Yep, caffeine on reaction time. Okay, so if you take 2 points of data on page 227, it looks very linear. And if you take 4 points of data on page 227, you can see that the table there is not as it would have seemed with only 2 points of data. So more data, the better. That's what we are going to focus in on there. Okay, so when should we do these bar graphs? Bar graphs should be done when we have a discrete variable like whether, let's see, whether they get um, the the they're in a sensation perception experiment, and you are going to give people a green maze to draw in and kind of complete versus a yellow maze. Okay. So green, yellow, those are the different conditions. Those are not a continuous variable. They're not something like time. They're not something like trial numbers. They're not something like milligrams of caffeine, right? They're either you got the green, did I say green, or the yellow, all right? And that would be a discrete variable, the independent variable. And so that would be done. The dependent variable would be on the left. Uh, I always forget X, Y axis. The Y axis, right? I think that's the Y axis. Sorry, it's been a while. Yep, the Y axis would be the dependent variable, and then the independent variable would be discrete, and it would be a bar graph on the X axis. Okay, so hopefully that's pretty straightforward, and why that happens would be straightforward as well. Okay, so I want to revisit really quickly with t-tests and ANOVAs when we're doing inferential statistics. And these will be the only inferential statistics that we will really talk about in this course. And so you're going to need to try to understand, again, how these are determined. Now, I'm not going to need you to do them. I just need you to understand how we get to generalizing our findings. Okay, so if you have a well-controlled experiment, which we talked about last chapter, right? So if you've used random assignment or matching or counterbalancing, okay, now if you have a well-controlled experiment, now your findings can be meaningful because there's no confounds, or you have decrease the amount or the effect of the confounds. And now we are going to use a t-test or ANOVA to tell us, can we generalize this to our population from our sample, our small sample, out to our population? If it's true here, what's the likelihood that it is true elsewhere? And we don't ever deal in absolutes. We deal in likelihoods. Is it statistically significant? We assume that we may have found this to be true, and there's a chance, this is the type 1 error, that what we found to be true here is not true in the population, but we can be some percentage, 95% in some cases, depending on how we do the statistic. We can be 95% confident that this is true. Okay, and that's what the t-test is going to show us. That's what the ANOVA is going to show us. How is it going to do that? It is going to measure the dependent variable. So remember that we always have multiple people involved in our studies for multiple data points, whether it's a between group study and we split them into two groups for two conditions, or it's a within group study and the same person is exposed to both conditions of the independent variable. In either case, there's not just one or two people in the study, right? There's 40 people in the study, right? And so you have these, let's say we have 40 people in a study, and we're going to randomly assign them into two groups of 20, or within groups we have 40 people in the study, and we are going to let them experience both levels of the independent variable. So in either case, we have, like in the between groups, we have these 20 people to compare data to with the other 20 people who have experienced different levels of the independent variable. And then within groups, you have 40 people to compare to themselves as they experience different levels of the independent variable. Are you with me? Okay. So in either way, you are comparing scores of one level of the independent variable to another level of the independent variable. Let me say that again. It's very, very important. In either case, the whole point of an experiment is that you are comparing one level of the independent variable to another level of the independent variable. And how do you do that? By measuring dependent variable, okay? So the results of one group who's experienced one thing to the results of another condition who has experienced another thing. And that's where the dependent variable gets measured. Okay, so in either case, you have this data set. Let's just pick a middle ground. Let's say that we have 30 pieces of data in one level and 30 pieces of data in the other level. Now, they're always going to be the same, right? Because whether you're comparing people against each other or to themselves, you kind of want that matched group. Doesn't have to be, but it's nice when it happens like that. So these groups, these two groups of 30, these two sets of data, let me say it that way. So we have 60 pieces of data, 30 and 30. They are going to have some amount of variation within those groups of data. So what if you gave one group of people a certain type of driver training, and then the second group of people a different type of driver training, and you wanted to measure safe driving was the dependent variable. Okay, so we have the safe driving scores of both groups, 30 in each side. Now here's what we want to know is the standard deviation of both groups. So you need to know what all the individual scores are, what the standard deviation is of each data set, and that information is going to get plugged into either a t-test or an ANOVA, and that that statistic is going to let us know whether this is generalizable. Now, what are they looking at? They're looking at the difference between groups versus the difference within groups. And let me say it a different way so it's not confusing. They are looking for the difference between the data sets. How much difference is there between the data sets with these two types of driving kind of lessons? How much difference is there in the dependent variable? Wow, this group over here that looks like they did way better than this group over here on average, right? It looks like that. Now we need to ask ourselves, but what is the difference within each data set? Maybe this one that looks so good has a ton of variation in it. So we had people on average, yeah, it looked like people did pretty well on average, but there are a group of people who scored really low, and then lots of people scored really high, and really nobody in the middle. Tons of variation, high standard deviation versus this group that on average didn't seem to do as well as this group, but they had this tighter data set where it didn't have the people who scored really low and people who scored really high. It had people scoring pretty well in the middle and quite consistently. Well, guess what? A t-test and an ANOVA is going to take all that into consideration. And while it may look like on average one group did better and even significantly better than the other group, it is going to tell us there is too much of variation in the data set to confidently say that you can generalize this to the population. Okay. Hopefully that helped. The t-test and the ANOVA help us generalize our findings to the population. They are looking at and comparing the variation within the data sets and across the data sets. And that's how an ANOVA or a t-test comes to this conclusion. Once you get into stats, if you haven't taken it yet, you will actually do ANOVAs and t-tests. All right. Well, I'm hoping that was somehow helpful to you as we get into the work this week and work towards a revision. If you have any questions, put them in Ask Questions here. And be sure to show up for our class this week so that you can get the help you need for doing a good revision. Thanks so much for your attention to this. Take care.